Hi, my name is Nicole Roberts. This is week 4 of microbiology. In this video lab, I'm going to be completing exercise 1, which is the methyl red and Vogue's Pro Scour testing. So I have all of my materials laid out here that is needed to complete the experiment. I have my MRVP broths. I have my E. coli broth tube. I have my S. Epidermidis broth tubes. So I'm just going to pretty much go ahead and follow the lab manual and complete this video lab. Okay, so let's go ahead and get started. So I've already disinfected my work area, and I've already labeled my my tubes. I've placed the droppers in the pipette. I'm sorry, I placed the two dropper pipettes in the disposable cup of alcohol, so that's there. So now I'm going to remove the lid of the E. coli MRVP broth tube and flame the lip of the tube to the flame. And here is my E. coli MRVP broth that I have labeled. Okay, I have my flame here. So I'm going to carefully insert the tip of the pipette into the active E. coli culture, drop a small amount of broth, and then I'm going to pipette 4 of those drops into the active culture, into the MRVP tube. Okay, so here is the MRVP broth for E. coli. And I'm going to take my active E. coli cultures. I'm going to disinfect this as well. Perfect. Okay, so 4 drops of the active E. coli culture into the MRVP broth tube. Okay. I said 4 drops. Okay, there's 4 drops of that. Okay, actually, I'm going to flame the lip of the MRVP broth and screw on the cap. Alright, so I've done that step for the MRVP broth for E. coli. So now I'm going to go ahead and do the same thing for the MRVP broth for the S. epidermidis. Okay, so I'm just going to get this out the way here. Okay, here is my active culture for S. epidermidis, and here is my MRVP broth. So I'm just going to make sure that I use aseptic techniques and make sure everything continues to be clean and disinfected. Okay. So I'm going to do the same thing that I did for the E. coli broth. I'm going to do that for S. epidermidis. I'm going to take 4 drops the active culture and pipette it into the MRVP broth. I'm going to screw on the cap. Okay, so I have pipetted my 4 drops of the active cultures into the MRVP broth. So I'm going to go ahead and incubate this in my incubation location, which is the cabinet in the hallway. I'm going to incubate these tubes for 48 hours, and then I'll go ahead and check on them again. And then that concludes my week 4 video lab for the methyl red and Bogue's Pro Scour testing. Thank you so much for watching.